Our next speaker is Dr. Yang Hansil from KAIST. So he's a very young, promising scientist. He graduated from KAIST and then did masters with Go Gyu-young 교수님 in KAIST, but went over to Rockefeller University to study with Elaine Fuchs, who's a pioneer in epidermal stem cell research. So he did a PhD with her in Rockefeller and did a very short postdoc with her and came back to KAIST in 2020, last year. So he's currently assistant professor in KAIST. He will be talking on the epigenetic regulation of hair follicle stem cell maintenance and differentiation. Without further ado, Dr. Yang.
Thank you very much for kind introduction, and thank you very much for organizing this meeting and inviting me here to present my research. As Chair mentioned, I just set up my lab last year, and today I want to share with you my research that I have done in Elaine Fuchs' lab last 6 or 7 years. So tissue regeneration is key survival mechanism in our life. So for example, in skin wound or bone fracture can be healed by the action of stem cells. So interestingly, in most mammals don't have this ability, but some salamander or other lower organism can regenerate their missing body part completely, like this. So beyond this tissue regeneration, the action of adult stem cells is key event for this tissue regeneration. So adult stem cells is the cells that can self-renew and differentiate into different functional cells. And in contrast to embryo stem cell, adult stem cell has to be long-lived through our life. So once tissue regeneration begins, the stem cells start to proliferate and undergo reactivation, specification, and terminal differentiation process. So we call this process as a stem cell lineage progression. So if this process is perturbed, there are many disease conditions can be caused, like cancer or degenerative disease can be caused. So our lab and my interest is skin epidermis. So in skin epidermis, there are two tissue resident stem cell population. So first one is epidermal stem cells that are located at the surface of our skin and produce skin barrier. And the other population is hair follicle stem cells that are invaginate into dermis site, and predominantly these cells regenerate hair follicle and produce hair. And if you focus on hair follicle stem cells, there are two subpopulations. One is long-term body stem cells, and the other one is short-term hair germ cells. So once hair cycle begins, these short-term stem cells start to proliferate and grow down and engulf derma papilla, which is specialized derma fibroblast population. And then these hair germ cells continue to proliferate and start to differentiate and move upward. And in fully regenerated hair follicle, there are active proliferation of these progenitor cells to produce hair. Actually, the speed is very fast, the 20 micrometer per hour. So our hair is generated. And then, after destruction stage, the hair follicle stem cell is degenerated and returning to the resting phase. Interestingly, during this hair cycle, the long-term body stem cells proliferate just briefly in the middle of hair cycle to replenish the stem cell pool. So hair follicle stem cells give rise to 8 different hair follicle lineage cells. And also when you have skin wound, the hair follicle stem cell can participate in epidermal wound-healing process. So this is clinically important because when you have skin wound, most of wound is superficial. The superficial epidermal stem cells and the upper dermal cells can be damaged. So to heal the wound, epidermal stem cells at the edge of wound site So you have to migrate this long distance. So it can cause the secondary immune infection and tissue degeneration. But in this case, the hair follicle stem cells that are located at the bottom of wound site can move upward and participate in wound healing process. So in clinic, the wound in scalp lesion in human heal faster than other body part. So also hair follicle stem cells, once hair follicle stem cells get oncogenic mutation, the hair follicle stem cell can give rise to cancer stem cells that sustain the skin cancer. So going back to the hair follicle regeneration, this is the resting phase of hair follicle, and through the hair follicle stem regeneration process, hair follicle, this big and long hair follicle can be regenerated. And if you focus on this hair follicle, there are 8 different hair lineage cells, and these 8 different cells are morphologically distinct, as you can see in EM image. And also they express different molecular marker, and their function is also different. So these inner 3 layers forms hair, and the other 5 layers forms supporting layer. So the question that I have in Elaine Fuchs' lab is when and where and how these hair follicle stem cell lineage choice is made. So there could be 2 possibilities. One model would be the spontaneous differentiation model. The hair follicle stem cells differentiate into different lineage cells at some point simultaneously. And the other possibility is the hierarchical progressive model. The hair follicle stem cell first give rise to multipotent progenitor cells, and the multipotent progenitor cells go to unipotent progenitor cells for each lineage of hair follicle. So to study these questions, hair follicle was a good model system because the stem cell and their progeny, the spatial relationship of these 2 stem cells and their progeny is well characterized. And also in mouse, hair follicle regeneration is physiologically synchronized. So because these 2 regions, I focused on to solve this question. To study this, we first analyze the proliferating cells during hair follicle regeneration. By EDU incorporation assay, we found that the cells that are closely located at the derma papilla is highly proliferating in early anagen to middle and the full growth phase. So this suggests that it should be there are active close talk between the hair follicle stem cells and their progeny at the bottom of hair follicle and the derma papilla. This active crosstalk will be important. So we wanted to answer the when and where these lineage activated, highly proliferative progenitor cells are specified into different 8 lineage cells. To study this, we utilize single-cell RNA-seq analysis. So we use 1965 PCR plate-based single-cell RNA-seq, the SMART-seq2 protocol. First, we divide the FACS purification strategy to purify the proliferative progenitor cells in each hair cycle. And we FACS purified these cells into 1965 PCR plate, and using SMART-seq2 protocol, which is more sensitive and more robust than the drop-based 10x genomics method. And also, using the bioinformatic analysis, we tease out the heterogeneity of these hair follicle stem cells and proliferative progenitor cells. So as you can see here, using this method, you can detect about 6,000 to 10,000 genes per cells. So this is 2 times or 3 times more than the 10x genomics method. To make long story short, I want to summarize our result. So first, in the telogen before the stem cell activation, we found that the short-term hair germ stem cells is already specified into 3 populations. And the population that are very close to derma papilla start to proliferate in the early stage of proliferation. Then this population goes to specify into first hair lineage cells companion layer, but this population remains in as a multipotent progenitor cells. And then layer stage, these multipotent progenitor cells give rise to two distinct hair shaft and IRS, two distinct hair lineage cells. And finally, in anagen 6, which is the fully regenerated hair follicle, there are 8 unilineage progenitor cells that are specially located just next to dermapapilla cells. And these progenitor cells rapidly proliferate and divide asymmetrically. The one cells remain in basal layer and one cells give rise to subobasal layer. And these subobasal cells move to the upward to make the columnar structure, the onion-like columnar structure of a hair follicle. So based on this result, we wondered, so how these hair follicle stem cell identity and their gene expression are determined and regulated. So actually, about 10 years ago, Rick Young's group suggests that the sulfur enhancers are determinant of cell identity and state. So compared to typical enhancers, the sulfur enhancers are the large clusters of active enhancers. So as you can see here, sulfur enhancers have the special regulatory function, the characteristics. The first, when activator concentration high, their expression, transcription activity is stably maintained, but when the specific concentration of activator, the transcription activity is switched like a switch. So using super enhancer characteristics, we can have on-off switch-like transition. So for using these, the many cells, including ES cells, some brain cells, adipose tissue, and the immune cells, their key transcription factors are associated super enhancers, and also these factors are important for establish the super enhancers. So we try to identify super enhancer network in hair follicle stem cells in vivo. So using H3K27 acetylation chipseq and mediator 1 chipseq and other key transcriptional chipseq in hair follicle stem cell in vivo, we were able to identify about 400 super enhancers and their key transcription factors, the SOX9, LHX2, the TCF or NFIB, and MFC1. So based on this, we want to study how these super enhancer network is changed during the hair cycle lineage progression, and how the diverse external signaling effectors, such as Wnt or BMP, Notch, or Sony Hedgehog signaling, converge on the chromatin to direct dynamic cellular fate choices during hair follicle stem cell specification. So today I want to show you two examples. The first one is hair follicle stem cell activation step. The other one is the IRS and hair shaft, the specification steps. So for this activation step, we did ataxic open chromatin region analysis because at that time there is no available single cell ataxic or single cell chipseq technology. So we FACS-purified the in vivo hair follicle stem cells and hair germ cells and compared their open chromatin profile. So as you can see here in CD34, which is key stem cell genes, so the ataxic open chromatin region is greatly reduced, but in COOX1, which is key differentiation genes in hair follicle, the open chromatin region is, the chromatin is opened in these hair germ cells, the lineage activated hair germ cells. So for the genome-wide level, so we identified about 30,000 ataxic peaks are specific to lineage activated these hair germ cells. And by motif analysis, we found the key, the differentiation transcription factors are enriched in these peaks. Importantly, we found the TCF and LEF1 motif in at the top of this list. So TCF-LEF1 is key regulator of Wnt signaling pathway. So Wnt signaling pathway is a very famous and very important signaling pathway in stem cell and cancer and many development pathway. So when Wnt ligand binds to Wnt receptor, the beta-catenin is stabilized and translocated into nucleus and to activate the gene expression. However, the beta-catenin has no DNA binding domain, so it needs the DNA binding signaling effector such as TCF3, TCF4, and LEF1 and TCF1. So first, we try to analyze the expression pattern of these Wnt effectors, TCF3 and 4, and LEF1 and TCF1. Interestingly, we found very exclusive expression pattern of these 4 Wnt effectors. So TCF3 and TCF4 is highly expressed in resting phase, the quiescent hair follicle stem cells when Wnt signaling is off. But when Wnt signaling is on, the TCF3 and 4 is turned off, and TCF1 and LEF1 is going up and continuously expressed. And also, one interesting finding was when the TCF3/4 was deleted in adult, you can see the precursor stem cell activation. However, when TCF3 and 4 is knocked out in the embryo, their follicle formation is lost. So based on these two different results, so we wondered the effect, the action of different Wnt effector TCF3 and 4 and LEF1, TCF1 during the hair follicle regeneration. So to answer this question, we did in vivo chip-seq analysis. So we did TCF3 and 4 chip-seq and H3K27 acetyl chip-seq for to test their the cis-regulatory region activity. And also we did chip-seq of LEF1 and H3K27 acetylation in the proliferated progenitor cells. So we found 3 interesting categories of these binding patterns of Wnt effectors. First, the first category shows TCF3 and 4 binding was replaced with LEF1 binding, but their activity is maintained in the stem cells and the differentiating cells. And these category peaks are associated with pan hair follicle, pan epithelial genes such as EGFR and DSP and these genes. And the second category is TCF3 and 4 bindings are lost in proliferated progenitor cells, and the activity is going down. So these suggest that these peaks are important for maintaining stem cell identity. So if they are lost, the precursor differentiation can be caused. And the most interesting category is the third category. So TCF3/4 bindings at the key differentiation genes, but they don't activate the gene expression. But when they are replaced with the LEF1, the other Wnt effector, the regions become active. So this indicates that there are dual function of TCF3/4 bindings in the stem cells. The first, the bindings activates the key stem cell identity genes. And second, the bindings bookmark key differentiation genes and facilitate LEF1 switch. So maybe this bookmarking or replacing this binding is support the fast and efficient differentiation of fast and efficient activation of differentiation genes. So to test if these possibilities is valid, we did one genetic deletion experiment. So we deleted LEF1 and TCF7 in hair follicle stem cells, and we also did hair plucking, hair regeneration model. Then in wild-type mouse, after hair plucking, hair follicle is regenerated and hair is recovered. But when we delete these two genes, the hair recovery didn't take place. And also, the TCF342 LEF1 switch is not happen, and also key stem cell factor SOX9 is still expressed. And also many key differentiation genes are not turned on. So this suggests that so during stem cell activation, the TCF342 LEF1 TCF1 switch is important. And we wonder the next stage of IRS and hair shaft specification. So after the LEF1 switch, the LEF1 and TCF1 is continuously expressed in these highly proliferative progenitor cells during the hair cycle. And also, when you compare the LEF1 peaks at these hair follicle progenitor cells and their superenhancers, we found that 70% of superenhancers have at least one LEF1 peaks. This suggests that LEF1 is master transcript factor for lineage progression in these proliferating progenitor cells. So question is that how these uniformly expressed LEF1 regulate the IRS and hair shaft lineage specification. So to answer this question, we tease out the LEF1 peaks based on the single-cell RNA-seq data. So we take the IRS gene signature and hair shaft gene signature from single-cell RNA-seq data and tease out the LEF1 chipseq based on their association with the gene signature. And we found that by gene motif analysis, we found that the LEF1 motif is common denominator in two populations, but the other motifs are differently present in these two populations. In IRS population, Notch, NFI, and GARA3 motifs are present, but in hair shaft, PRDM and HOXY13 and FOXQ1 or FOXP1 motifs are present. So we wonder if these differences are functionally relevant or not for the hair lineage specification. To answer this question, we focused on the one key transcription factor, the COOX1. The COOX1 expression is uniformly expressed in IRS and hair shaft lineage genes. And interestingly, COOX1 has two superenhancers. The one is proximal and one is distal. So we took out the key cis regulatory element from the enhancer A here, the proximal enhancer, and enhancer B, and the distal enhancer. And then we made the reporter construct. Then the reporter is EGFP is driven by the enhancer region, this enhancer A and B peak, and the PGK driven HTB RFP as a transduction condition. Control, sorry. So after making this lentivirus, we injected lentivirus at the E9.55 yolk sac. So by injecting this lentivirus in the yolk sac, we can transduce skin epithelia specifically. Then in the adult, we analyze the EGFP expression and RFP expression. Interestingly, when we cloned out this enhancer A peaks, so we found this EGFP expression is overlapped with GARA3, which is IRS lineage marker, but the COOX1 B enhancer is now overlapped with GARA3, so is inner layer of GARA3, which is hair shaft lineage. So when we analyze the binding pattern, binding motif of these two enhancer fragments, we found LEF1 is co-bound, these two enhancers, but there are different patterns of binding patterns. So in enhancer A, IRS enhancer, there are RBPJ, GARA3, and NFI motif, and enhancer B, there are SMED1, HOXY13, and FOXQ enhancer motif. So among these motifs, we focused on RBPJ and SMED1 motif because RBPJ is a downstream of Notch signaling pathway and SMED1 is downstream of BMP signaling pathway. To test this functional relevance of these two enhancers, we mutated the left one binding site and RBPJ binding site in the enhancer region, and we found that the enhancer activities was going down, like here and here. And also when we analyzed RBPJ knockout hair follicle, we found that the GARA3 positive IRS lineage was not formed normally. But the HOXY13 positive, the hair shaft lineage was normally present. So this suggests that the left one, together with RBPJ, is important for IRS specification. And how about the other, the lineage, the hair shaft lineage? So we did similar experiment. We mutated left one binding site and SMED one binding site at the enhancer region, and we found the enhancer activity was going down. And also when we analyzed BMP receptor 1 knockout hair follicle, we found that the hair shaft lineage genes are specifically and uniquely downregulated compared to IRS lineage genes, which are not changed. So in summary, so using hair follicle stem cell lineage progression model, we found that how external signaling cues converge on chromatin to change their cell identity and stem cell lineage choices. In resting phase stem cell transcription factor, together with the TCF3 and 4, activate stem cell genes. Upon lineage activation, when UNT signaling is active, the TCF4, 3 and 4 binding is switched with left one and TCF1. And then later stage, the left one is work together with NOTG and RBPJ signaling to specify and activate IRS specific genes. But in other lineage, the left one is work together with phosphosmed1, the BMP phosphosmed1 to activate the hair shafted specific genes. So with that, I want to thank my previous mentor, Elaine Fuchs, for her great mentorship and her great support. And I also want to thank all Fuchs members for their support and their great suggestions. So as I mentioned, I just opened my lab, the Laboratory of Stem Cell and Tissue Regeneration at KAIST. So with great student, hopefully next time I can share our own data. So thank you, and I'm happy to take questions.
Thank you very much, Dr. Yang. So we have 5 minutes more for discussion. So this wonderful talk is open to discussion now.
Yeah. Professor Yang, it's pretty interesting. And I wonder that like your 8 different lineages and when they are committed. And so thinking about like those cells are from the bulk stem cell and the matrix cell, and then they committed their like a decision and their destiny. And I wonder whether those like commitment are like a stochastic or any deterministic based on their topological system. So I wonder if you have any kind of like idea, and then would you share that things to me?
First of all, actually one of our previous comment is transplantation, but we couldn't do that transplantation assay. The transplantation assay shows how this, if the cell fate is deterministic or plasticity, or we couldn't tell that, but we think that the lineage specification is determined by the spatial relationship between the dermapapilla. So when the hair germ cells proliferate and engulf DP, this engulfment induces the spatial distribution of the hair follicle stem cells, the progenitor cells and dermapapilla. The upper part becomes hair shafted progenitor cells, and the lower part becomes IRS progenitor cells. So that suggests that this location, the spatial relationship is critical for the lineage specification.
So along that line, so what kind of factor can decide that those kind of spatial, like the location? So certain cells should be upper and then certain cells should be lower. And that means like, is there any kind of like a driven factor which can be higher or lower, depending on the population or any other?
Yes, that's a great question. So I think that would be determined by their location in the telogen phase. So in the telogen phase, the cells that are located at the periphery of hair germ because this is epithelial cells. Epithelial cells have to move they cannot move individually. They have to move together. So the cells that are located at the peripheral region of the hair germ cells become the cells that are located at the downside of hair bulb. So they should become the hair IRS lineage cells. And the cells that are located at the center of hair germ part, they have to be at the upper part of hair bulb. So they will give rise to the hair-shaped lineage cells, I think.
Thank you. Actually, I had a related question. So what I understand is that from the bulge stem cell, the hair follicle stem cells, depending on how it moves, migrates, that they epigenetically change the factors to become the differentiated lineages, right? So is there any way of kind of switching this? Like if you can mark a cell which was supposed to go down and make it go up, and does the epigenetic marks change? I mean, do you know how you can approach that, or is there anybody who has done this experiment?
That's a great question and very interesting question. Actually, to do that, we need very specific genetic marker to label and target the specific population. But unfortunately, I don't think anyone have that kind of specific genetic driver. So that would be a very interesting question.
In lower species, you can actually like laser ablate one cell and see the effect of the fate of that organism. Like I'm just wondering whether you can have a kind of ex vivo system to kind of laser ablate out one of the bulge cells and see if those migration actually affects. So it was just a crude idea, but I thought it was very interesting. So related to what you have said, that spatial, temporally controlled way of epigenetic regulation was quite interesting. Yeah, so next question I had was, so you have given really solid data on WNTs and the TCF lefts on how they actually regulate, master regulators for this hair follicle stem cell lineages. But, you know, I mean, these TCFs and lefts are actually critical factors for the embryogenesis itself and also in T cells. So what makes the difference between hair follicle cells and T cells, or even the very primary embryogenesis? What is the difference? Is it based on the different WNTs, or is it the combination of every signals coming together?
That's very fundamental, very interesting question. I think the most important determinant is epigenetic landscape. So I think the binding of TCF effectors can be regulated by their binding, their chromatin environment, whether they are open and whether they are closed, and whether they are associated with other histomarker marker. I think the key difference would be the epigenetic landscape.
Thank you very much. So yes, thank you very much, Dr. Yang. Right.